Hello everyone and welcome to video tutorial 12. You should now know all about the first unit, cell biology. Well done for getting this far. If you're struggling with cell biology, remember you can go back and watch the videos of the parts you're struggling with as many times as you want. Today we are going to be moving on to multicellular, which is unit 2. Multicellular is broken down into 6 key areas. The first key area is producing new cells, and that's the key area that we will be looking at today. The second is controlling communication. The third is reproduction. The fourth is variation and inheritance. The fifth is transport systems in plants, and the sixth is transport systems in animals. Today we're going to look at key area 1, producing new cells. If you want the worksheets to go with the video, remember to pause the video and go to 2 multicellular, 1 producing new cells, 2 consumables and activities, 1.1 cell division. The PowerPoint is found in PowerPoint and is called 1.1 cell division. So today we are going to be learning about cell division. Our learning intention is to learn to state that mitosis is cell division, identify that mitosis is needed for growth and repair, describe the different stages of mitosis, state the importance of cell division producing an exact copy of a diploid cell, and state where cell division happens in plants. To be successful, you will be able to create an accurate model of the stages of cell division. You will be able to say and put the stages of cell division in order, and you will be able to correctly answer the cell division questions. Before we start learning about cell division, let's think about how we have become 75 trillion cells from one cell. Also, why is it important that these cells get all the same genetic information? And lastly, what diseases are caused by cells not reproducing the genetic information correctly? So pause the video and see if you can write down an answer to the questions. The answer to the first question is that one cell becomes 75 trillion cells by cell division, which is also called mitosis. The next answer is that it is important that all cells get the same genetic information so that no genetic information is lost. The last answer is that cancer is caused by uncontrolled cell division. Well done if you managed to get the answers correct. If you didn't, we're away to go talk about this in more detail anyway. Cell division is the means by which an organism increases its number of cells. Can you think of any reason why we might need to increase the number of cells we have? Pause the video and write down what you think. The reason that multicellular organisms need to increase their number of cells is for growth. Multicellular organisms need to grow from a cell into an organism, for example, a zygote to a human. Also, multicellular organisms need cell division for repairing any damaged cells or for the replacement of dead cells. For example, if you cut your finger, then the cells need to be replaced. This is an important note to make, and you need to remember that cell division, also known as mitosis, is used for growth and repair. So make a note of that. From the previous unit, you should be able to label the parts of the cell and describe the function of A. You can find the diagram in the consumables in activities 1, chromosome and nucleus. So pause the video and try to label it before I go over the answers. A is the nucleus. The function of the nucleus is to control cell activities, including cell division, which is also known as mitosis. And you should have learnt that in the first unit. The nucleus stores all of the genetic information, so C would be your DNA, which is your genetic information. And the DNA is around about 2 meters long, and it has to be wrapped so that it can fit into the tiny nucleus that you can't really see by the naked eye. You've got to look at it under a microscope with dye. So that is really, really tiny. So the DNA must be wrapped up into something called chromosomes, which is B. Well done if you managed to answer the last question correct. Now we would take a note on chromosomes. Chromosomes are thread-like structures that are composed of DNA and contain genes. They are found in the nucleus. You need to know the structure of a chromosome. So the chromosome is made up of 2 identical chromatids, and they are held together by a centromere. So you've got your 2 sister chromatids here. That's one of the sister chromatids, and that is the other sister chromatid, and they are held together by something called a centromere. So you would take a note of that. Here are microscope images of same chromosomes in different cells. You need to know that each species of plant and animal has a characteristic number of chromosomes called its chromosome complement. Every normal human body cell contains 46 chromosomes arranged as 23 pairs. A cell with two matching sets of chromosomes is said to be diploid. You can remember this as di means two, and we'll come back to this when we go to look at reproduction. So you take a note of what chromosome complement means. The number and appearance of chromosomes in the nucleus is displayed in a karyotype, just like the one that you can see in the photo. This can be used to check for any abnormalities in chromosomes, which could mean you have a disease like leukemia if your chromosome 9 and 22 are mutated, or if you have an extra chromosome at 21, then you'd have Down syndrome. A picture of a normal human karyotype can be found in Consumables and Activities Free karyotype diagram. This is just another picture of a karyotype. The sex chromosomes are positioned in number 23. So if it's XX, it's for her, and it would be two long chromosomes. If it's XY, it's for him, and it would be a long and a short chromosome, like you can see in this picture here. Note that all of your chromosomes are arranged in 23 pairs. So there's 46 of them, but they're arranged in 23 pairs, and this would be their diploid number. Normal human body cells are referred to sometimes as somatic cells, and they're diploid, whereas sex cells, the sperm and the egg, they are haploid. This is because they only contain 50% of the genetic information. Sex cells, or gametes as they're called, are described as being haploid, as they only contain one set of chromosomes. You can remember this by half meaning half. Gametes are produced from the normal diploid cell, so you've got your 23 pairs of chromosomes here, and then each gamete receives one copy of each chromosome from every pair of chromosomes. So gametes only have one set of chromosomes and are said to be haploid. So you'll notice that the two have went down to one, so it's only got one set of chromosomes. All organisms have a different chromosome complement. For example, an onion has 60 chromosomes in its normal diploid cell, and so it must have 8 chromosomes in its haploid sex cells. See if you can pause the video and work out the rest of them. So the answer would be that the cabbage has 18 in a diploid cell and 9 in a haploid cell. The bee would have 8 in a diploid cell and 5 in a haploid cell. The frog would have 24 in a diploid cell and 12 in a haploid cell. Wheat would be 24 in a diploid cell and 12 in a haploid cell. The cat would be 38 in a diploid cell and 19 in a haploid cell. And the humans would be 46 in a human diploid cell and 23 in a haploid cell. So well done if you managed to answer them all correctly. We would now take a note on mitosis. Cell division is the process of dividing one parent cell into two daughter cells with exactly the same genetic information. Mitosis is the division of the nucleus. In mitosis, the number of chromosomes within the nucleus is firstly doubled. Then the chromosomes are divided between the nuclei of two daughter cells produced by cell division. This ensures that the diploid chromosome complement of the cell is maintained so that the cell can function correctly. It is important to watch the video link. You can pause this video and watch it, or you can go back to it at the end. Mitosis produces diploid cells and occurs in carefully controlled stages. So let's have a look at the stages in detail now. You need to be able to know each stage so that you can put them in the correct order, fill in a stage if it is missing, or you might get asked a 4-mark question asking you to describe each stage. Therefore, it is important that you go over the stages, write them out and say them. Even make a song about them, or just make sure you can answer questions on them. In stage 1, the nucleus contains long uncoiled chromosomes. Each has been replicated to form an X-shaped structure composed of two identical chromatids, which are held together by the centromere. The chromosomes cannot be seen under a microscope at this stage. So you would want to take a note of stage 1. It is important to note that as long as the chromosomes remain attached to each other, the two identical copies of the chromosome are known as sister chromatids. They are only known as chromosomes when they are separated. In stage 2, the sister chromatids condense and tightly coil to become short, thick, and visible under a microscope. We can see that the chromosomes are still in the nucleus of the cell at this point. So you would take a note of stage 2 now. In this stage, you'll notice that the nucleus has disappeared. So for stage 3, we would write down that the nuclear membrane dissolves and then the spindle fibers appear. So they appear from these poles. Here are the spindle fibers that I'm highlighting in red. The sister chromatids then line up at the equator, which is the center of the cell, just like you would have the center of the Earth. And they attach to the spindle fibers at their centromere. So your sister chromatids are attached at the centromeres, and that is what you would note for stage 3. So the nuclear membrane dissolves, the spindle fibers appear, sister chromatids line up at the equator of the cell and attach to the spindle fibers at their centromere. In stage 4, the spindle fibers contract and separate the sister chromatids from each other. The sister chromatids, now referred to as individual chromosomes again, are pulled to opposite ends of the cell, or you can refer to the opposite ends of the cell as the cell poles. So as you can see, the sister chromatids getting pulled away from each other by the spindle fibers contracting. So you need to take a note of stage 4. In stage 5, a nuclear membrane forms around each set of chromosomes. Remember, the separated chromatids are now called chromosomes. So you take a note of stage 5. In stage 6, the cytoplasm splits into two new diploid genetically identical daughter cells are formed. So remember to go back, write down all of the stages and learn all of these different stages off by heart. You need to know that cell division in plants happens differently because plants have a cell wall. This means that the cytoplasm cannot split and it cannot be pinched. So instead, a new cell wall has to be made in stages 5 and 6. In plant cells, mitosis can only happen in meristems, in plants' root and shoot tips. So mitosis can only happen in the root and shoot tips in places called meristems. See if you can pause the video and fill in the blanks to compare cell division in both animal and plant cells. You can find a diagram in consumables and activities for a cell division. So try to fill it out before I go over the answer. So the answers are that in animal cells, the nucleus divides and then the cytoplasm divides and then you get your two genetically identical daughter cells formed. Whereas in plant cells, the nucleus divides, then a new cell wall is formed, and then you get your two genetically identical daughter cells. So well done if you managed to fill that out correctly. Let's look over the keywords that you should now know. So pause the video and try to fill in the table which can be found in consumables and activities for C mitosis before we go over the answers. So the answers are that one of the pairs of threads which make up the chromosome during mitosis is the chromatid. The two chromatids of a chromosome are joined here would be your centromere. The region of the cell around the middle would be your equator. Fibers which attach to the centromere responsible for moving the chromosomes to opposite ends of the cell would be your spindle fibers. And the term used to describe the new cells produced by mitosis would be your daughter cell. So well done if you managed to get all of these terms correct. If you didn't, then you might want to go back to the start of the video. A common question that often gets asked in exams is why does the daughter cell have to be genetically identical to the mother cell? To answer this question or anything similar, the easiest thing to say is that so no genetic information is lost. So whenever you get asked that question, make sure you're right so no genetic information is lost. Another common question that might be asked would be to state the number of chromosomes present in this cell. Now, to work this out, you would have to, first of all, count the number of chromosomes in the cell. So you try to do that and pause the video before I tell you the answer. So the answer would be 1, 2, 3, 4. So there's 4 chromosomes in that cell. Then you get asked, state how many chromosomes will be present in each of the 2 cells produced by the process. Now we know that the cells have to be genetically identical to their parent cell and to each other. So therefore, if this one has 4, then the 2 daughter cells will also have 4. So the answer would be 4 for the first one and 4 for the second one. So a top tip is to note that the parent cell will always contain the same number of chromosomes as the daughter cell. For example, if the parent cell contains 3 chromosomes, the daughter cell will contain 3 chromosomes. If the parent cell contained 2 chromosomes, the daughter cell would contain 2 chromosomes. The next question is, why is mitosis important? So pause the video and try to write down why you think mitosis is important before I go over the answer. So the answer is that mitosis is important because it maintains the diploid chromosome complement and it ensures that there is no genetic information lost, so that each daughter cell has all of the genes needed to carry out the cell's function. So usually in class we do a little partner activity where you'd both stand up, and before you could sit down you must say all of the 6 stages of mitosis accurately to your partner without looking at any of your notes. To check to see how well you've done, pause the video and see if you can put the stages into the correct order before I go over the correct order with you. So the correct order would be that first of all it would be number 3, so each chromosome replicates itself to become an X-shaped structure composed of 2 chromatids which are held together by the centromere. Then it would be number 2, so sister chromatids become short, fat and visible, so you can see them under the microscope. Then it would be number 1, the nuclear membrane dissolves, spindle fibers appear, sister chromatids line up at the equator of the cell and attach to the spindle fibers at their centromere. Then it would be number 5, the spindle fibers contract and separate the sister chromatids from each other. The sister chromatids, now individual chromosomes again, are pulled to opposite ends of the cell, which is also referred to as the cell poles. Then it would be number 6, so a nuclear membrane forms around each set of chromosomes. And lastly, it would be number 4, so the cell's cytoplasm splits and 2 new diploid genetically identical daughter cells are formed. So well done if you managed to get that all correct. If you didn't, you might want to go back and watch the start of the video again. You should watch the mitosis song by pausing the video and going to the link, or by going back at the end. You should also do the same with the mitosis crash course by pausing it and watching the link, or going back to the link at the end. This will just make sure that you know and understand everything. We mentioned earlier that a disease caused by the uncontrolled cell division is cancer, and you need to take a note of that. So uncontrolled cell division causes cancer. The problem-solving questions that you sometimes get asked in cell division are, for example, a cell divides every 20 minutes. How many cells would be produced from one original cell at the end of 2 hours? So to get this answer, you would first have to convert your 2 hours to minutes. So to do that, you would do 2 times 60, which would give you 120 minutes. Then you would divide 120 minutes by how often the cell divides. So that would be 20 to see how many divisions that took place, which would give you 6. Then you can do it in 2 ways. You can either do 2 to the power of 6 in your calculator, or you can do 6 2's together as shown, so the answer should give you 64. So as you can see, convert 2 hours to minutes, so you get 2 times 60 equals 120 minutes. 120 divided by your 20 minutes to see how many divisions, so there was 6 divisions. So then you do 2 to the power of 6 in your calculator, or you can do 2 times 2 times 2 times 2 times 2 times 2, which is the same thing. So there's 1, 2, 3, 4, 5, 6 2's, and then it gives you 64. So well done if you managed to follow that. So we'll try another one. So this time it says a cell divides every 30 minutes. How many cells would be produced from one original cell at the end of 4 hours? So you guys pause the video and try to do it yourselves. So first of all, we need to convert our 4 hours to minutes, so 4 times 60, which gives you up to 240 minutes. Then we need to divide it by the minutes to find out how many divisions there was. So 240 divided by 30, so that gives you 8 divisions. So you can either do 2 to the power of 8, or you can do 2 times 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 2. So there should be 8 2's there, and it will give you 256. So again, pause the video and try to answer the question before I go over the answer. So a cell divides every 30 minutes. How many cells would be produced from one original cell at the end of 3 hours? So the answer would be that you'd convert your 3 hours to minutes, so it'd be 3 times 60, which would give you 180 minutes. And then you'd want your 180 minutes divided by your 30 minutes to see how many divisions there was, so it would give you 6. So then you want to do 2 to the power of 6 to see how many cells we get. So 2 times 2 times 2 times 2 times 2 times 2, so there will be 6 2's there, which gives you 64 cells. So last time, so try to answer a cell divides every 20 minutes. How many cells would be produced from one original cell at the end of 1 hour before I go over the answer? So the answer would be that you'd have to convert your 1 hour to minutes, so that'd be 1 times 60, which would give you 60 minutes, and then we need to see how many divisions. So we do our 60 minutes divided by our 20 minutes, which would give us our 3 divisions. So you would either do 2 to the power of 3 in your calculator, or you could do 2 times 2 times 2, which would give you 8 cells. So well done if you managed to get them correct. If you didn't, then you can go back to the first calculation and watch it from there. Another type of problem-solving question is a bacterial cell has an initial concentration of 1000 cells per centimeter cubed. Each cell divides once every 30 minutes. Calculate how long it will take for the concentration to become greater than 1500 cells per centimeters cubed. So pause the video and try to work this out yourself before I go over the answer. So to get the answer, we know that 1000 will double each 30 minutes. So 1000 to 2000 to 4000 to 8000 to 16,000, that is 4 times. So that is 120 minutes, but we want hours, so we would then need to divide it by 60, which then would equal our 2 hours. So well done if you managed to get that correct. So now let's do a little true and false quiz to review our learning. Humans have 44 chromosomes. Would that be true or false? So the answer would be false, because humans have 46 chromosomes. So the next question is, a chromosome is made of 2 chromatids. So is that true or false? It would be true, so well done if you managed to get that. So during mitosis, a parent cell divides into 3 daughter cells. Would that be true or false? So the answer would be false, and the reason being is because a parent cell would divide into 2 daughter cells, not 3. If cell division is not highly controlled, this can result in cancer. Is that true or false? So the answer would be true. Cell division is needed for growth and repair, so do you think that's true or false? The answer would be true. Now you can complete the activities below to review your learning. So watching the Amoeba Sisters videos, where the link is below, doing the extension questions that you can find in activities and consumables, along with the worksheets 4 to 8, doing the multicellular exam practice questions page 21 to 22, doing the past paper questions on cell division, which are found in 4 past papers for key areas. Finally, you can model the stages of cell division with sweets like strawberry laces or pipe cleaners, and you can write an explanation of each stage beside it. This will help you to review your knowledge and help to make sure that you understand the work. We have now come to the end of the lesson, so you should now be able to state that diploid cells have 2 matching sets of chromosomes, state that a human body cell has 46 chromosomes, describe how this is maintained during mitosis, describe each stage of mitosis, state that mitosis is needed for growth and repair, and state why it is important that exact copies of the cell is made. So well done if you can confidently do all of that. If you can't, then you need to watch this video again until you can. This is the end of this video tutorial. Woohoo! Thank you for listening.